Good morning. My name is Brittany Lasseigne, and I'm going to talk to you today about building a platform for precision animal model selection and drug repurposing prioritization. I'd like to introduce the Center for Precision Animal Modeling, a recently funded U54 with the goal of becoming a national resource to efficiently and cost-effectively analyze pathogenicity of gene variants identified in patients with rare disorders and to produce variant-specific models to pursue disease mechanisms and targeted therapeutics. Our co-directors are Brad Yoder and Matt Might, and they also lead the coordinating center, which is responsible for providing organization and management, developing outreach and recruitment, and coordinating access. My role is as the co-director of the bioinformatics section. So how does the center work? So for patient variants for which UAB or the external community, anybody watching can submit a variant, please go to the CPAM gateway. And the idea here is that folks would like an animal model to assess the impact on phenotype or have a model for other studies, including future assessment of drug repurposing candidates or other drug candidates. You can nominate them through this portal. And this is then evaluated by the co-clinical section, which is led by Dr. Korf, Might, and Kraus. They have set up the CPAM gateway here and a nomination form. They collect information about the variant in the patient and ensure that the information is correct and complete. From this, they generate what's called a phenopacket with a program called Phenotips. Phenopackets are an open standard for sharing disease and phenotype information with the goal of improving the community's ability to understand, diagnose, and treat disease. And it links the phenotype description with disease, patient, and genetic information. In addition to these responsibilities, the clinical and co-clinical section is also responsible for IRB approval of CPAM clinical enrollment and coordinating iterative phenotyping of human and model systems. After generating the phenopacket, it's shared with the bioinformatics section, which is led by Dr. Liz Worthy and myself, and I'll dive a little deeper here given my role in the audience. But generally speaking, our team is responsible for building the computational platform that CPAM runs on for precision animal model selection and drug repurposing prioritization. So practically speaking, this means the BIS is building tools and pipelines to aid with all the intricacies associated with analyzing, curating, and aiding in variant review and prioritization. So we review, analyze, and reclassify variants according to ACMG guidelines, and when necessary, by working with the clinical and preclinical core. We also work on phenotype definition and exploration and patient animal model type, genotype, phenotype comparisons. But this goes even deeper to gain insights for variant case prioritization and selection. So we do deep dives into model organism conservation and variation, where the variant falls with respect to a given gene annotation, work understanding what cell and tissue gene and protein expression is typical, predicting the impact on the protein and providing rich information and interpretation regarding what is known about that gene or the gene product's role in the gene. Additionally, for the original variant review assessment, we perform literature and knowledge database driven predictions for drug repurposing, and for variants selected to make a model, we also provide omics driven predictions. For a select set of models, we generate RNA-seq or other omics data in my lab and do an even deeper workup that includes analyses like transfer learning with public data. Overall, though, our goal is to identify or predict and prioritize therapeutic candidates from literature and knowledge database mining, use of molecular profiling data from public data, patients, or model organisms, and predicting and prioritizing therapeutic candidates. We're particularly focused on drug repurposing because of the potential to bypass long and expensive drug development if possible. I look forward to future opportunities to discuss this pipeline in more detail with the community. There are several other additional critical roles for the bioinformatics section, including using best software and data management practices, well-documented, robust, and reproducible systems and tools, and coordinating and providing oversight for the entire data life cycle. This includes data assessment, QC, storage, integration, visualization, and model organism genotype, phenotype data, experimental metadata, and patient genotype and phenotype storage. Finally, in coordination with others in CPAM, we're dedicated to aiding and distributing the community with the knowledge and resources generated, including packaging of data for model services and resource dissemination. So as you can imagine, it takes a whole team. And one of the things that I think is really cool about the bioinformatics team is we have software developers, engineers, cell biologists, genomicists, and data science, along with the overall interdisciplinary CPAM team to help us make sure the analyses we're performing are relevant. Here are the team members from Dr. Worthy's lab and from my own lab. So once we've generated a variant review report with all of this rich information for assessing variant impact and model selection, we then meet as an entire team across all of the sections to discuss gene and variant prioritization and assignment to appropriate models based on criteria that can broadly be described as pathogenicity, suitability, drug potential, and patient impact. So the rest of the team you haven't met yet are pictured on this slide, as well as several staff and students who also contribute. So the disease modeling unit, which is everyone on the slide, is responsible for production of in vivo models of gene variants, as well as developing and implementing new technologies and enhancing capacities. The resource and services section, which is a subset of that team and is responsible for acquiring and documenting strains and materials, characterizing, expanding, and distributing resources and outreach to the community. Additionally, the RSS provides access to other unique services that include candidate drug target prediction, cell line and iPSC model generation, other omics, and specialized phenotyping. So these models, characterizations, drug prioritizations, et cetera, are then available back to the research community, and something that makes the center unique is this outreach and team building. So far, we've evaluated, or we've had over 50 projects enter the pipeline for evaluation, and we have several models that are being generated. So I want to give a end here and give a big thank you to the entire CPAM team, as well as my lab, which is pictured here, and a thank you for your time and attention today.